This is Lesson 10B, which is Part 2 of Transgenic Approaches for Drought Stress Tolerance. In the previous lesson, we talked about lines that had been engineered with transcription activators and LIA proteins. And in this lesson, we'll talk about strategies using antioxidants, compatible osmolytes, and an RNA chaperone mechanism. Let's look first at the antioxidant strategy. Now, drought stress, like several other abiotic stresses, results in accumulation of reactive oxygen species, and these damage membranes and other cellular components. As you're aware, plants contain various antioxidants that naturally counteract these effects. But what if the concentration of those antioxidants could be increased transgenically? Would that provide higher levels of stress tolerance? Well, several enzymes have been engineered in plants to increase the antioxidant level. And it raises the appealing possibility that antioxidant-enhanced plants could be good for the plants by giving them better tolerance to heat and drought and cold, but also could be good for the people who consume the plant products. This slide lists some examples of the antioxidant transgenic strategy, and you can find the references in Ashraf. The gene for superoxide dismutase, SOD, has provided a higher level of stress tolerance when overexpressed in alfalfa and potato. An ascorbate peroxidase gene was engineered in tobacco chloroplast and resulted in tolerance to osmotic stress. A gene for vitamin E synthesis called VTE1 was engineered in tobacco, resulted in increased drought tolerance. This was associated with reduced electrolyte leakage and less lipid peroxidation. These all sound kind of encouraging. However, I will point out that only the alfalfa SOD study involved field testing, and in that case it was positive. The compatible osmolyte strategy was based on the hypothesis that if you increase the concentration of osmolytes in plant cells, you would increase the ability for those cells to osmotically adjust and therefore withstand dehydration better. Some of the common compatible osmolytes are glycine betaine, proline, trehalose, and mannitol. And there have been projects where each of these have been overexpressed. You can read about those in Ashraf if you choose. But I'll go into some detail about a case of wheat engineered with MTLD gene from E. coli. This was published by Ababa and colleagues in 2003. Some background is that mannitol is a sugar alcohol that occurs naturally in many plants, but not in wheat. Researchers at Oklahoma State University developed a transgenic drought-tolerant wheat that accumulates mannitol in plant cells. I'll show you what constructs they used in just a minute. Previous research had shown that transgenic mannitol accumulating tobacco and Arabidopsis had improved drought tolerance. It was later found that this effect may not be due to osmotic adjustment at all because the quantities of mannitol accumulated are so small. So the effect may really be due to antioxidant activity or protection of membranes. This is a simplified view of the mannitol synthetic pathway. Starting from fructose 6-phosphate, if you have the mannitol 1-phosphate dehydrogenase enzyme, which is encoded for by MTLD, then you get the conversion to mannitol 1-phosphate. And nonspecific phosphatases present in any plant cell will then convert that to mannitol. Now note that fructose 6-phosphate normally goes to seed storage carbohydrates, and so if you introduce MTLD into a plant, you don't want to have a super active enzyme because that would take away from your potential yield by detracting from the seed storage carbohydrates. Well, at Oklahoma State, the constructs they used were three. They had a construct that would accumulate the transgenic protein in the cytosol. So that consisted of the BAR gene, which is the selectable marker for herbicide tolerance, the MTLD gene driven by a constitutive UBI1 promoter terminated by the NOS sequence. There was a construct that accumulated the protein in the chloroplast. So in addition to the selectable marker, it had the same construct as the cytosol version, except it had this chloroplast transit peptide, which would direct the protein from the cytosol into the chloroplast. And then finally, what's sometimes called an empty vector. It was the selectable marker without any of the trait gene, without the MTLD gene present at all. This is from the ABUBA reference. If you look at the dwarf plant first, most of their transform lines were like this. They were short, they were sterile, they had twisted heads, and they had very high mannitol levels. But a few of the transformants had normal phenotypes and only 1/3 to a half as much mannitol. So that middle pot is of a transformant with a more or less normal phenotype compared to the control on the right side. So it was those transformants with only a reduced amount of mannitol that were continued in the study. But how did these plants perform under low moisture? Well, the photo in the pot on the left shows the control, no stress. And the two pots on the right were both under stress. The center pot was without the MTLD gene, and the right is with the gene. So under stress, the MTLD containing plant doesn't look as vigorous and normal as the unstressed plant, but quite a bit better than the plant that doesn't have the MTLD gene. So this was somewhat encouraging, and it caused quite a bit of publicity when it was published. My colleague Scott Haley, who's the wheat breeder at Colorado State, and I got excited about this. We contacted the Oklahoma researchers and we arranged to grow their transgenic lines in a field trial. And we ended up doing this for 3 years under wet and dry conditions. So this is a photo of what our trials look like. We evaluated 7 transgenic lines. These were T6 and T7 generations. Randomized complete block design because it was a fairly small trial with 3 reps. Wet and dry treatments, and this was the 2007 version. We had furrow irrigation where both treatments received irrigation just until flowering, and then we cut off any irrigation from the dry treatment. Now, one thing I want to point out in this trial is some of the precautions that have to be taken when you grow an experimental transgenic line out in an open field situation. First of all, we surrounded the trial with winter wheat, so that vegetative bushy stuff you see in the foreground is winter wheat that was not vernalized. And so it wouldn't flower, there would be no danger of cross-pollination to that material. The plants in the background, those are the transgenic lines, spring wheat, and they're at full heading in this picture. We had to surround the field with the fallow area where nothing was planted. We had to guarantee that there was no wheat or jointed goat grass within 330 feet. We had to ensure that no wheat or other small grain would be planted in this field for 2 years. So it does put a restriction on crop rotation schemes. And then we had to monitor for volunteer plants for 2 years after the trial. So quite a cumbersome procedure, but there's more. Dedicated equipment is a requirement, or at least a recommendation, and so we did a lot of work by hand that normally would have been done by machine. So on the left is harvesting the trial with hand sickles, and then we took the bags over to this ancient thresher that we found in an old machine shed and resurrected, and threshed our plants that way. We had to meticulously inspect the thresher afterwards to make sure there were no seeds left behind. I always told my students, I'll give you $1 for each seed that you find after the cleaning was finished, and usually they would find a couple of seeds. The bottom picture is a Peebles jumping meadow mouse. Now, what is that doing here, you might wonder? Well, before we planted the trial, we had to guarantee to USDA that this transgenic wheat trial would not harm in any way the Peebles jumping meadow mouse, which is a threatened species, and it does grow in Colorado in the general vicinity or the region where the trial was conducted. However, there were good maps of where its habitat was. It was far away from where the trial was planted, and so we were able to demonstrate that it wouldn't be a threat. But just an example of the different hoops you have to jump through to grow a transgenic trial, and that is certainly one of the reasons why there aren't more field studies done on transgenics. The results of this MTLD trial are shown here, with the orange bar being the dry treatment and the green bar being wet treatments. The bars indicate the yield in tons per hectare according to the scale at the bottom. So the parental cultivar was Bob White, the top line, and then there were 4 events that were expressed in the cytosol, 2 events expressed in the chloroplast. There was the empty vector, which is transgenic but does not contain the MTLD gene, and then a Czech variety. This is a conventional variety from North Dakota, which has a reputation for drought tolerance. So, the empty vector, what's going on with that? We would expect that the empty vector would be pretty similar to Bob White, but it's not. It was very small and low yielding compared to the other lines. So we think that this was just something that happened in the transformation or the tissue culture process that just ended up in a weak plant. You can see that Bob White did pretty well. The only thing that exceeded it was Reader, which was the conventional Czech. None of the transgenics did as well under the wet treatment, and all but one was poorer in the dry treatment. Now, not all these differences were significant, but we can say that at least none of the transgenics was significantly better than Bob White under either treatment. Couple that look relatively good, though, would be the PTA 2, 110, 115, and 118. There was no statistical significance in the difference between the cytosol and the chloroplast expressing lines, but numerically the cytosol lines were better. Now, when we look at things in terms of yield reduction between the well-watered and the moisture stress treatment, it looks a little different. Bob White on the left has about a 42% reduction. The best of the transgenics was the 118 line, only about 28% reduction. So it looks like there is some hope that some of these transgenics may have some degree of tolerance. Reader, by the way, looks like the best. That's the conventional line, only about 21% yield reduction. So why did this happen? Why were none of the transgenics better than the parental cultivar? Well, we came up with three potential explanations. One was that the transgene was lost through multiple generations of seed increase. Remember that these were T6 and T7 plants. So somehow the gene may have been eliminated, excised itself, or some other process, or maybe some non-transgenics got mixed in during the seed increase. We conducted some PCR analysis for the presence of the gene in 25 plants of each of the lines, and we did see variable rates of the transgenic presence, from as low as 68% in one of the lines up to 100% in 118. And as you'll recall, the 118 was the best performer. However, there wasn't really a good correlation between performance and percent of gene presence. Another explanation is that the gene had been silenced in all or some of the transgenic lines. And this is consistent with work of Elavarti, who also worked on this material. And he saw some degree of silencing between the T3 and T4 generations. And then finally, maybe the gene is present and maybe it is expressed, but the mannitol is not accumulated, or its accumulation is not effective in protecting the plants from yield reduction under the conditions of this study. And that's something that Ashraf also points out in his review, that it's still not clear whether mannitol has a protective effect or not. So conclusions of that study were, first, contrary to previous findings, in controlled environments, we did not detect improved grain yield under drought stress in this set of lines in 8 field environments. I should mention that in addition to 4 Colorado environments, we also had 4 environments in North Dakota. Secondly, when compared as a group, the differences between lines designed for the protein accumulation in the cytosol versus the chloroplast were not significant. However, individual cytosol lines had the best performance among the transgenics, including a smaller percent reduction under drought. Lines that appeared to be the most promising were 118 and 115. Finally, evaluation under more severe moisture stress or evaluation of additional MTLD lines might provide more promising results for potential incorporation of the gene into cultivars. Now I'd like to talk about what may be the first engineered drought-tolerant crop to be released. And this is a corn event jointly developed by Monsanto and BASF. It's known by its serial number as MON 87460. It has been touted as being ready for the market by 2012. I'm not sure if that has slipped or not. The transgene involved is CSPB, which encodes a cold shock protein from Bacillus subtilis under the control of a constitutive promoter. Now this is a very interesting mechanism. The gene produces an RNA binding protein with chaperone activity, the result being that gene expression is maintained under stress conditions. The Monsanto researchers evaluated a number of different genes or events with cold shock proteins. Some were from E. coli, and the gene is termed CSPA, and the B. subtilis protein is CSPB. These were put into hybrid backgrounds, and this data is based on multiple managed stress sites. So the top group in the table is the CSPA group from E. coli, where the control was 6.38 tons per hectare. The mean of the CSPA group was 6.68, which was a 4.6% increase. But there were two specific events within that group which looked quite good. One was 30% higher and the other 18% higher than the control. Looking at the CSPB group, the mean of the event group was 7.5% higher than the control. The two outstanding events were 11 and 20% higher. So it was actually the CSPB ZM event 1 that the researchers continued to work with, and that was the one put forward for deregulation in the event that's currently being evaluated. This chart shows yield results when that CSPB ZM1 event was put into 3 different hybrid backgrounds and evaluated across a range of western dryland sites. So not the very managed, controlled situation, but more or less farmer field type situations with uncontrolled weather patterns. The gray bars show the transgenic, and the white bars is the parental control. So in all cases, you have from about a 5 ton per hectare up to somewhat above 5.5 tons per hectare. So about a 10% increase. Now what's striking to me is how similar and stable the results are in 3 different hybrid backgrounds. And apparently these results were pretty stable across the drought stress sites as well. So that's exactly the type of situation you would like to see before releasing transgenic hybrid. This event is especially being targeted to the western Corn Belt, which includes eastern Colorado. The expected improvements are a 5 to 10% increase in yield under moderate drought stress, not severe stress, with no yield penalty under well-watered conditions. Well, when I first heard this, I thought 5 to 10%, that doesn't seem like so much. But a grower pointed out to me, well, if you're growing 200 bushels per acre, get a 10% increase would be 20. And if corn is going for $6 and up a bushel, well, that's significant and important. And so for corn, it seems like that degree of yield increase would be sufficient. Some questions would be, will the same gene be effective in other grain crops? Now, Monsanto recently bought a wheat seed company, Westbred, and the speculation is that they will try out this gene in their wheat cultivars as well. But will the same level of yield improvement be sufficient? Wheat yields in Colorado are very low, only about 30 bushels per acre. So an additional 3 bushels, maybe not worth the cost of transgenic seed. Anyway, we'll have to wait and see. We may see this on the market as soon as next year. Of the various examples I've pointed out about experimental transgenic lines, it's really been a mixed bag of results. Some have been quite promising and warrant further investigation, and others less so. I am certainly not negative about the prospects for transgenic drought tolerance, and I think that work should be encouraged and should continue. but I also think it's important to be realistic about some of the limitations of the approach. So here is my subjective and personal list. You may have your own concerns. First, will a single transgene be able to improve tolerance to drought that occurs at different times and severities? As we've discussed various times in the course, there are many different scenarios for drought, and can we manage expectations of a farmer so he doesn't expect that regardless of severity and time of drought that drought-tolerant cultivar will give outstanding yield. Secondly, transgenes are not always stably inherited and may experience gene silencing. That's why you need to start out with many transgenic lines in order to get one or two that are stable and high yielding. Third, the genes that may offer the most dramatic improvements in drought tolerance, transcription factors, involve the most complex responses and regulatory issues. You've seen that genes like DREB1A affect a whole slew of different things. So each one of those responses is a regulatory red flag that would need to be investigated. Next, the transfer of drought tolerance to wild relatives of crops may have negative effects on ecosystems. This really depends on which crop is being talked about. It is an issue, as we've said before, with jointed goatgrass from wheat, but it wouldn't be an issue in the US for corn because we don't have wild relatives of corn here. It wouldn't be an issue for soybean, but it would be an issue in the places where those crops do have wild relatives. Finally, field testing of experimental transgenic lines is expensive, burdensome, and inherently risky. There have been cases where experimental lines somehow got into the food supply or threatened the food supply. It prompted recalls and very large expense for the people involved. So in summary, plants transformed with genes for antioxidant enzymes, such as SOD, ascorbate peroxidase, and enzymes for vitamin E synthesis have shown enhanced stress tolerance, but few have been field tested. Increasing the concentration of compatible osmolytes has been the goal of several research efforts involving glycine betaine, proline, trehalose, and mannitol. They have resulted in improved drought tolerance in enclosed environments, but not in the field. A drought-tolerant genetically engineered corn from Monsanto may be released commercially soon. And finally, field testing of experimental transgenic plants is expensive, logistically challenging, and risky. So this is the end of the unit on transgenic plants.